Good day, everyone. Welcome and thank you for joining us from across the globe. My name is Chen Cai. I'm President and Chief Medical Officer of TWI Biotechnology. It is my absolute privilege to welcome you to our global webinar, Unveiling the Future of Epidermolysis Bullosa Care, Clinical Update and Path Forward on AC203. At TWIB, our guiding philosophy is that every patient and their family deserve a better life. Today, we bring together an extraordinary panel of world-renowned physicians and scientists to discuss a devastating disease, explore critical patient needs, and share the latest updates on our clinical efforts to bring tangible hope to the EB community. I will be your host for today's session. Before we start the presentation, I want to quickly go over a few housekeeping items. First, we'll have a Q&A session at the end. Please feel free to drop any questions you have into the Q&A box at the bottom of your screen throughout the presentation. We'll address as many as we can during the Q&A session. You'll notice that your microphones are muted, but we highly encourage your participation. Next slide, please. We have an excellent agenda lined up for you today. Following my brief opening remarks, our senior consultant, Dr. Michael Chen, will provide an EB simplex disease overview and a review of our clinical trials. Next, we are incredibly honored to have Professor Johann Bauer, Director of EB House Austria, join us to speak deeply on dystrophic EB, the clinical reality, pressing patient needs, and an ongoing named patient program. We will then transition into a panel discussion featuring all of our speakers, alongside Professor Amy Paller from Northwestern University, before opening up the floor to your questions in a live Q&A session. Next slide, please. Our panel brings together decades of deep translational research, clinical trials, and direct patient care. For myself, as a pediatrician by training with over 10 years of clinical practice and research, and a background at the Taiwan Regulatory Authority, CDE, I know firsthand how vital rigorous science is to bring rare disease therapies to light. Next is Dr. Michael Chen, Senior Consultant at TWI Biotech. Dr. Chen brings over 30 years of translational drug development expertise in rare skin and neurological disorders. He also serves on advisory boards for Cure Congenital Muscular Dystrophy and the Association for Creatinine Deficiencies. Joining them are Professor Bauer, a globally recognized pioneer who has authorized over 150 peer-reviewed publications. He is widely recognized for his significant contributions to EB research and novel therapeutics for rare skin diseases. His proof of concept trials of topical dioxazine for EB simplex were instrumental in advancing the clinical development of AC203. Finally, Professor Paller, a legendary investigator in pediatric dermatology who has led more than 200 clinical trials. She's also a principal investigator for NIH-funded research focusing on EB and inflammatory skin diseases. We are incredibly grateful to have this level of expertise dedicated to today's discussion. Next slide, please. Next slide, please. Let me share a brief snapshot of who we are at TWI Biotech. We are a clinical stage pharmaceutical company focused on accelerating science to deliver hope. Our core development strategy leverages the US FDA's 505(b)(2) regulatory pathway by building upon known established mechanistic and safety profiles. We are able to drastically reduce development risks and accelerate timelines to get therapies to patients faster. Strategically, we target two key therapeutic pillars, dermatology and the CNS. This focus is anchored by our highly secure IP portfolio. Next slide, please. This brings us directly to our lead asset, AC203. AC203 is a first-class topical diacetyl 1% ointment designed specifically for EB simplex, a condition for which there's currently no approved drug. We are leveraging an active ingredient with a 30-year established human safety profile from oral use, but formulating it into a novel topical ointment that acts as a topical IL-1 beta inhibitor to reduce keratin aggregates. Recognizing its profound potential, regulatory authorities have granted AC203 orphan drug designation in the US, EU, and Taiwan, alongside rare pediatric disease and fast track designations by the US FDA. It's currently the fastest developing EB simplex drug globally, having successfully entered a pivotal phase 2/3 study, with previous data demonstrating rapid onset, significantly improving skin lesions within just 2 months. We are incredibly excited about the path forward. With that context, let us dive into the clinical depth. I will now hand the floor over to Dr. Michael Chen to walk us through the EBS disease overview and our trial landscape. Michael, please.
Thank you very much, Dr. Chai here. So it's a great pleasure for me to be here, and thank you very much for the TWR Biotechnology for this opportunity here. My name is Yumo Michael Chen, and I'm a senior consultant, for the company here. So today, what I'm going to do is first, I would like to go over the entire the diacetyl program for EBS, which is developed by TWR Biotechnology in Taiwan here. So I will first, give a quick overview of the disease, and then describe how topical diacetyl will work for EBS. And then finally, I would like to present some of the clinical data for the EBS studies here. Next slide, please. EBS, epidermolysis bullosa simplex, or simply EB simplex here, is a rare skin disease causing skin fragility here because of the pathogenic variants in genes encoding very key important structure proteins in the epidermis here. Even it's a rare disease, it affects more than almost 500,000 patients globally here. So clinically, the major symptoms for EBS is blister formation here, and the blister actually are recurring. That means actually the blister forms, heal, disappear, but reappear. Could be in different locations or the same location at a different time here. It causes pains, itching, and severely and negatively affect the quality of life of the patients here. So if you take a skin biopsy and then you look at it under the microscope here, which is in the middle part here, the skin has a very organized structure. You can see the epidermis, a thin epidermis at the top, which is separated by the much thicker dermis by the basement membrane here. So in EBS patients here, because the fragility is at the epidermis, in particular, the basal keratinocyte, the cell that actually immediately above the basement membrane here. So quite often, they break apart here. So this is a very typical histology feature in the EBS here. And many genetic variants, could cause EBS here, but predominantly, the most prevalent genetic variants are the keratin genes here, the K5 and K14 in the keratinocyte here. And they are very important structural protein to maintain the integrity of keratinocyte in the epidermis here. As you can see from the right-hand side of the figures here, they form a network here to provide a mechanical support for keratinocyte and therefore the skin here. So if you look at a real keratinocyte image here, you can see the keratin network, or what we call the intermediate filament network here. So you form a very mesh network extending from the nucleus, which is the black hole in the middle here. Next slide, please. EBS can also divide it into different subtypes based on the clinical severity of the blisters here. So currently, there are 3 major subtypes here. Localized, as the name applies here, the blisters tend to be mild and actually localized to predominantly hands and feet, as you can see in the one of the pictures on the left-hand side here. And there are also intermediate type and the severe types, which the blisters tend to occur over the bodies here. So collectively, they are called generalized EBS here. Many genes can cause EBS here, but as I mentioned, the majority of the patients here are result from the pathogenic variants in the keratin 5 and keratin 14 genes. And interestingly, there's a very good genotype and phenotype correlation here. That means actually, if we know the location of the mutation in the keratin 5 or keratin 14 here, quite often we can correlate with the severity, whether this is a mild form or whether this is a severe form here. So next slide, please. The active ingredient of the product is diasterin, which is a structurally related compound that quite often found in many natural compounds, such as alloy here. And actually, you can see the name AC203 because in industry, quite often we use a code name, to represent the program here. So in this case, this is AC203, which is the topical diasterin, 1% ointment here. And TWI Biotechnologies is using a repurposing strategy, as Dr. Chai mentioned here. Quite often, there's another name is called repositioning here. So how it works is, for diasterin, the oral form has been approved to treat osteoarthritis in many countries, in particular, for example, in Europe and in Asia here, based on the property that it inhibits the IL-1 beta production. And the IL-1 beta is a pro-inflammatory cytokine that involved in many diseases here. And over the decades here, there have been a number of studies, pioneered, for example, by Professor Bao, one of our guests here, that show actually the IL-1 beta could play an important role in EBS here. So the repurposing strategy is to change the formulation from oral to a topical into a 1% ornament here, and then to treat the EBS, which is currently TWI Biotechnology is conducting a pivotal study called the EB-SHOW study here. Next slide, please. The reason why actually topical diasterin would actually be useful to treat generalized EBS is based on 2 very important pharmacological activities that are highly relevant to EBS here. The first, the anti-inflammation, that actually has been mentioned, is the property to suppress IL-1 beta, a major pro-inflammatory cytokine. As you can see in the picture here, quite often in the EBS patient, in particular the severe type EBS patient, besides the blister here, they also manifest inflammation. You can see around the blister here by the red arrow on the left-hand side here. In addition, diasterin can also reduce the keratin aggregations here. You can see from the picture, this is an electron microscopy picture on the left-hand side here. So typically, a normal keratinocyte, they will form a network of the keratin filament here. But when the keratin network is disrupted, quite often it collapses into an aggregate. You can see the black electron-dense material that actually is the pathological hallmark in the EBS here. So, in addition, a topical formulation here can deliver enough drug directly to the skin or to the blister here without the systemic side effect, which quite often associated with the oral formulation. In this case, the systemic effect is mostly diarrhea. Next slide, please. Here's the example of how diasterin actually can reduce the aggregates in the EBS cells here. So actually, on the right-hand side here, those are actual EBS keratinocytes isolated from the patient here. Without the treatment here, you can see a very characteristic of the punctated structure representing keratin aggregation here. So those dot-like structure denoted by the yellow arrow here. So with treatment with diaziran here, all these keratin aggregates began to disappear here. And we can also quantify the extent of the keratin aggregation here in a separate experiment that are conducted by TWI Biotechnology in collaboration with a local university here. As you can see it, in the normal keratinocyte, the one blue here, there are only very few cells showing the keratin aggregation here. But if we are using a EBS cell isolated from a patient here, up to 30%, the red bar here actually can show keratin aggregation. When treatment with the diaziran here, the number of keratinocytes showing the keratin aggregates are reduced to less than 10%. Next slide, please. Next slide, please. Yes, thanks. So by suppressing the IL-1 beta, the pro-inflammatory cytokine signal linked to inflammation, and also by reducing the keratin aggregation in the keratinocyte in the epidermis here, we believe actually diaziran can reduce the blister, the severity and the number of the blister, and offer better protection for skin against mechanical stress and trauma here. So in the middle figure here is our keratinocyte with all this aggregation and inflammation here. Without any treatment here, the keratinocyte will eventually breaks apart because of the mechanical trauma, inflammatory injury here. When treating with the topical diaziran here, it reduces the inflammation, the microenvironment become more favorable, and then it also reduces the amount of the keratin aggregation here. Even without a proper keratin network here, the cell becomes stronger and more resistant to stress here. And that's one of the reasons why we call our current pivotal study of the EB show here to offer better protection for the skin. Next slide, please. And the application of topical diaziran is very easy. The strength is 1% ointment here, and the patient can apply once a daily. And the method of application is very convenient here. So you just apply a very thin layer over the blister here. The patient can also apply the ointment to a clear skin area that may be prone to blistering based on the patient's personal experience here. There are no special procedures such as dressing, bandage required for the application here. So far, the diaziran, based on the previous clinical study here, demonstrates an excellent safety profile in EBS patients with very minimal skin irritation and almost negligible systemic exposure here. So the product comes with a 25-gram tubes, which is enough for up to 50% of the body surface area for an adult and 80% of the body surface area for an infant. Next slide, please. So now I would like to present the clinical data for EBS study here. But first, I would like to just briefly, go over the developmental history of diaziran program here. Actually, it spans decades, almost 30 years, since the 1990s here. And it's a collective effort from academia, industry, patient advocacy group, and regulatory authorities here. The genetic defects or the genetic pathogenic variants was discovered in the early 1990s here. And I have the very honor opportunity to be actually involved, in the discovery of the EBS along with Amy here. I still remember the good old time, with Amy in Chicago at that time here. And then during the last 10 years here, there have been a number of important works, in particular conducted by Professor Bauer, Dr. Wally, to demonstrate the role of the IL-1 beta in EBS. In addition, there are other PIs, Joyce Tan, they are also involved, trying to understand the pathogenesis of EBS here. And so far, there are 5 phase 1 clinical studies to support the safety of topical diasterin here. And this is part of a very long, tedious process required by FDA regulatory agency to demonstrate the safety of the product. And there are also 4 completed phase 2 study to support the safety and efficacy study. And today I'm going to present the data from 2 of the most recent studies here. And so far, currently, the sponsor, TWI Biotechnology, is conducting a phase 2 and 3 pivotal study called the EP Shield study to support the registration effort. And during the last 10 years too, the sponsor also actively talking to the regulatory agency, to guide the clinical program and to prepare for registration. Next slide, please. This is one of the first studies conducted by EB House under Professor Bauer and Dr. Wally here. At this study here, in this study, they recruit severe EBS patients with keratin K5 and K14 variants in 17 patients aged from 4 to 19 here. In this study, they treat the patients 4 weeks with either the diasterin, topical diasterin, or the placebo here, and then followed by 3 months with no treatment. They look at the end of the study here, they asked the question, how many patients can show reduction of at least 40% reduction in the blister number here. As you can see, at the end of the treatment, 4 weeks, almost 90% of the patient actually achieved this goal, whereas only 14% in the placebo group did. Even after 3 months, when the treatment stopped, actually the difference between the treatment or the previous treatment versus the placebo was still pretty impressive. And this is one of the picture, actually. You can see it, one of the patient here showing a very dramatic improvement in the number of the blister from the baseline to 4 weeks here. Next slide, please. This is the second study, actually the last study conducted a few years ago, co-developed by TWI Biotechnology and another company in the US called Castle Creek Pharmaceutical here. The study was called the delivery study here. And basically it's a phase 2 randomized placebo-controlled study, enrolled more than 50 EBS patients. The difference was actually at that time enroll all different EBS subtypes. That means from the localized to the severe one, and then the pathogenic variants could be keratin or other non-keratins here. And then they treat the patient for 8 weeks, followed by another 8 weeks of safety follow-up with no treatment here. And at that time, we also developed a new clinical assessment tool called IGA, Investors Global Assessment, to assess the average overall severity of EBS lesion at a particular time point here. This is a 5-point scale based on the clinical characteristic of EBS lesions to determine the overall severity of the EBS here. So you can see from the picture here, a score of 0 represents almost no blister. It's a clear skin here, whereas a score of 4 represent a very severe blistering in the patients here. And the success criteria based on the IGA score is if the patient come in with a score of 3 and 4, and then at the end of the treatment, the score reduced to 0 and 2, that means it's at least 2-point reduction here. It represents successful treatment. Next slide, please. And we'd like to show you 2 examples of the patients. This is a 7-year-old patient with severe EBS here. And when he came in to the clinical site here, he had an IGA score of 3 and the BSA area of the EBS blister of 6.3%. And at the end of the 8-week treatment here, the IGA score reduced to 1 and the BSA to 1.3%. So far, the safety profile based on the delivery study was excellent. Most drug-related side effects were local and mild here, and mostly related to skin discoloration, burning sensations, itching, and about only 20% of the patient receiving diasterin topical reported side effect here. And actually, we published the delivery results a few years ago. in 2023. Next slide, please. So this is another example. This was a 5-year-old girl, also with a severe EBS. She came in with an IGA score of 3 and a BSA of 8%, and then by the end of the treatment, the IGA score was reduced to 1 and the BSA to 0.5% here. You can see the picture, the improvement of the blister over the 8 weeks. Next slide, please. So here's the result of the IGA success from all patients here. So the delivery study enrolled about 50 patients, 26 receiving diastereon, and 28 receiving diastereon, and 26 receiving the placebo here. So you can see the patient receiving the diastereon, they show a steady and consistent improvement over the 8 weeks here. So by 8 weeks here, the number of the patients receiving the topical diastereon, about 40%, they achieved the IGA success based on the score of the 0 and 1 here, whereas in the placebo group is 23% here. So the difference was about 16% here. And one thing quite interesting, we noticed that because the cohort enrolled all types of EBS here, so we look at actually different subtypes here and then how they respond to the diastereon treatment. We noticed actually the patient with the severe EBS patient with a pathogenic variant in keratin 5 and 14 seems to respond to the treatment at a higher rate relative to the placebo here. So the figure on the right-hand side here, you can see those patients, about half of the cohort here. So by the end of the week 8, the end of the treatment here, those with the diastereon treatment show a 46% success. 46% show the success in the IGA, whereas 15% in the placebo achieved the success. So the difference was become 30%, almost double than all the cohort here. And the same thing for the intermediate EBS and also the severe plus the EBS, about 30%. So basically double the success rate, from all the cohort here. So altogether, with the EB House study and the delivery study here, it actually provide the basis, for this pivotal EBS study, the EB Shield study. Next slide, please. So next, yes. So the EB Shield study here basically is very similar in study design with the delivery study here. The patient also received treatment 8 weeks and then followed by 8 weeks no treatment, safety follow-up. And at the end of the Part A study here, all patients enrolled will receive AC203 for 6 months here to evaluate the long-term safety and efficacy of topical diastereon here. And so far, the study enrolled 80 patients here with severe or intermediate EBS with a confirmed keratin K5 or 14 variants here. And this is by far, in terms of the number, here's the largest EBS study ever conducted so far. And the primary endpoint will be the IGA score based on the 0 and 1 criteria I described earlier with a 2-point reduction from baseline. In addition, the secondary endpoint here, we also look at the change in the percentage of BSA lesion, and also the change in the pain and itching score, as well as the quality of life survey and other clinical assessment tools such as the EB Darcy score here. So next slide, please. And this is a global, truly a global study here, and that actually across 4 continents here. And we'd like to thank you, all the PIs involved in the study, the family, the EBS patients here for their active support. Next slide, please. And just a breakdown, of the number of the site and number of the patients here. So approximately, there are 3 major regions: the North America, Asia, and Europe here. It's about 1/3, 1/4, roughly, in terms of the site number and also the number of the patients here. So it's just truly global. Next slide, please. So I would like to actually end this part of the talk, with the update of the status of the EB-Shield study here. So far, the study, we enrolled 81 patients here. And overall, the dropout rate was very, very low here. And this is very amazing that given a clinical trial such as more than a half-year trial, usually, the dropout rate could be high due to many, many reasons here. And again, I would like to thank you for the support, for the patients and the PI here. So far, the overall safety profile was consistent with what we have observed in the earlier clinical study here. It is highly favorable. So far, the top-line data are expected to be available by the end of this year here. And meanwhile, TWI Biotechnology is actively talking to different regulatory agencies, to discuss the trial data and what are the next steps, possibly for registration. I would like to end the talk here, but the story does not actually end here, because we also believe, based on the mechanism, what we learned from the clinical study here, topical diastereon could also offer other potential in other skin blistering disease or wound healing disease here. And Professor Bauer here will actually will present, his latest data and experience regarding topical diastereon for treatment of dystrophic EB. Thank you very much for your time and attention. Thank you.
Yes, thank you very much, Dr. Chan. We move to dystrophic epidermolysis bullosa. But before we go into the depths of the data that we have on this subject, let me say a few words on the history of the whole story, because it's, it's an interesting developmental story from one side academia and on the other side of pharmaceutical companies, how they work together and how they develop new medications for the benefit of our patients. EB House Austria, which is kind of the core of this whole story of the diastereon topical ointment story, has been founded in 2005 in Salzburg in Austria. And we started as a very small unit. We had a clinical unit, the research lab, and the academic unit where we were providing education for medical personnel. And so one of the strands in research was to look into epidermolysis bullosa simplex. And soon after first 2 or 3 years, Dr. Wally came to me and told me they have found a very interesting molecule expression pattern in EBS cells versus normal keratinocytes, and this was already IL-1 beta. And so we looked into the databases, and that's how we came about diastereon, because it was already described to inhibit IL-1 beta. That was in 2010, and then we went on for a small study with 5 patients initially, looking into the armpits of the patients and trying to see if there is any effect from that topical preparation where we thought it might work. And there was effects. There was a reduction of blistering. And so we went on to the further studies that were already alluded to by Dr. Chan. And so this story kind of progressed very nicely for the patients. Now we have ample data for EB simplex, but let's have a look into dystrophic EB. Next slide, please. Yes, so for those who are not so familiar with dystrophic epidermolysis bullosa, it's another major subtype of epidermolysis bullosa. About 25% of the patients are suffering from this disease, and this is genetically very uniform, because we only see mutations in the type 7 collagen gene. Either there are stop codon mutations or deletions or insertions making up the recessive type of dystrophic EB, or there is missense mutations on one strand of the protein making up the subgroup of dominant dystrophic EB. Major clinical hallmarks in these patients is also blistering, as it is in EBS, in simplex. But in addition to that, there's major chronic wounding. So there is wounds that are staying there forever. They are staying there for months or even for years. They do not close. And even if they close, then they close with a scar. And so in the long run, patients are covered with a lot of scars on the skin. They have mitten deformities between their fingers, and in the long run, and very early in their life, they suffer from a high risk of becoming or acquiring aggressive cutaneous squamous cell carcinoma, which are very hard to treat. Next slide, please. And here's some clinical pictures from this disease, from dystrophic EB. Globally, there should be around 1,000 patients affected by this disease. And you see here in the chart to the right, as opposed to simplex EB, the level of cleavage is right in the dermis. In dystrophic EB, that's where the collagen 7 protein is actually located in the basement membrane. When there's a blister, it could heal. So there is acute recurrent wounds. You see this in the lower panel of the chart. But there is also long-standing wounds that are staying there for weeks or months, and then they are not closing. And you can imagine that there is. Excuse me. And there are complications like there could be infections in the wound. There is extensive fibrosis if the wound doesn't close. And in the long run, as I mentioned already, there is the risk of squamous cell carcinoma. Next slide, please. There is a few examples in the literature that point to L1 beta also in dystrophic EB, and mainly in recessive dystrophic EB. That's our research, but also research from other people in the field. They have analyzed the situation in the patients, in the wounds of the patients, and in the serum of the patients. And also in this subtype of EB, L1 beta plays a role apparently. L1 beta is increased in the serum. L1 beta is increased in the wounds. And in addition to that, we have also analyzed that in the first years in 2010 or so. There's also an increase in MMP9, matrix metalloproteinase, proteinase that is actually leading to a degradation of the collagen network. So these are two factors that are known in recessive dystrophic EB, and they could be amenable to therapeutic intervention because those two factors might lead to a delayed reepithelialization of the wounds occurring on those patients. Next slide, please. And you see here these data. There's data from a Spanish group to the left, Gregorio. They have analyzed the pores of hypomorphic collagen 7 deficient mice, and they see an increase in those chronic wounds of L1 beta and also of L1 beta secretion. Then there is older work from 2015 from a group in Italy, Anicchiarico et al. They have analyzed in serum of dystrophic EB patients various cytokines and have seen that there's also L1 is increased in dystrophic EB. And to the right, that's the work also from the EB House Austria. In 2015, we have analyzed the expression of various factors in pseudosyndactylia of those patients, meaning that that's the fissures that they have and mitten deformities on their hands where they lose their fingers. And in those areas of the hands, there is also an increase of L1 beta. Next slide. And that's a very recent work from our laboratories, from Sonja Dorfer, from Dr. Wallis' group, where she has looked more closely into that issue of L1 beta and also MMP9, because we know that MMP9 is a target of L1 beta. So if L1 beta is increased, it is implicated that MMP9 might be increased also. And we have proven this. And you see here to the left, we took whole cell lysats from patients with RDB, from keratinocytes from patients with RDB, and we subjected them to treatment with diasterin in cell culture. And you see that there is a modest, but there is reduction of MMP9 expression in Western blotting, and also in the supernatants, MMP9 is reduced. And you see that this is also statistically significant, this reduction with diasterin. And a second assay we did within this series of experiments to the right. This is a matrix degradation assay, and you kind of seed keratinocytes, in this case RDB keratinocytes, into a well. And this well is covered with a matrix. And then you let the keratinocytes basically there for a couple of days, in this case 72 hours, and then you judge where they have been moving to, and this is kind of a measure for the matrix degradation that is occurring with the keratinocytes. And if you follow those keratinocytes, you see the blue line after 72 hours. That's the normal movement of the keratinocytes, of the RDB keratinocytes. And if you add the diasterin, the movement of the keratinocytes is strongly inhibited. So they stay basically within the violet circle, meaning that there is a reduced matrix degradation when these cells are treated with diasterin. Next slide. So in total, that's our current hypothesis, how diasterin might work even in RDB. In wound healing, we know that it's suppressing L1 beta inflammatory signaling. That's pretty clear, but we also showed that there is a decreased expression and a segregation of MMP9 in RDB keratinocytes, and therefore we think there is a less matrix degradation, so there is better matrix if you treat the keratinocytes and the patients in the long run with diasterin. That would lead to a better reepithelialization and wound closure in those patients. Next slides. And this led us to our first named patient trial. This is our first patient that we have been treating once daily with diasterin on the back. And you see the day 1 and day 28 in this case. And there's a nice reduction of the big wounds in this patient on the shoulder, but also in the long run, also on the back, where the mother then switched also to treat the patient when she saw that it's working nicely on the shoulders. You see here on the back, those big wounds, they are nearly closed in this patient. Next slide. So together with TWRB, we are working at the moment on named patient programs. So we want to treat more patients with a local preparation with AC203, with that diasterin preparation, patients suffering from recessive dystrophic EB, and seeing how they fare, if there is any new side effects, how the safety profile is, and specifically how the wounding situation is. And next slide, please. This is one of the very good examples. At the moment, we have 4 patients treated. This is a fascinating example for me, actually. This you see here to the left, this is a lesion that has been there for 4 weeks. It stayed there, didn't move. And then it was treated with Diasterin for 1 week, and it's basically gone. So this was really fascinating. So certainly this is just one patient, one of the 4. And the other 3, we saw very good results also, but not that improving the skin. But certainly we will carry on studying this preparation in our inpatient program. Next slide. Yes, and so this ends the presentation for recessive dystrophic EB, and now we start with the panel discussion.
Thank you, Michael and Johan, for those excellent, highly informative presentations. We will now transition into our panel discussion. It is my pleasure to welcome Dr. Amy Paller to the stage, who joins Dr. Chen, Dr. Bauer, and myself for this segment. We have a lot of ground to cover today regarding the landscape of EB care, so let's dive right into our first question. Next slide, please. From a clinical perspective, what are the most critical unmet needs and disease burdens currently facing patients with EBS and RDEB? Additionally, what degree or type of treatment improvement would you define as clinically meaningful for these patient populations? Dr. Paller, I would like to invite you to share your comments first. Thank you.
Sure. Well, I do want to point out that EB simplex and recessive dystrophic EB do share their inflammatory profile and wound healing issues, but I do want to mention that they are somewhat different. So the big problem in EB simplex is more the skin fragility, the recurrent blisters which heal, but then they just keep blistering in these affected areas. As opposed to in recessive dystrophic EB, you've got the fragility, but also this delay in healing that Dr. Bauer was just talking about. And it's nice to see that there's evidence that combating that inflammation may be helpful in both, even though there are some differences. I want to emphasize that it's not just the healing, the fragility, but it's also the fact that these patients are in pain and are very itchy for many of them. And we think that a lot of this relates, again, to the inflammation as well. So unmet needs are different in these different patient populations, but certainly just focused on the wounds, but also what affects their day-by-day life and quality of life, the itch, the pain that are associated with these inflammatory skin areas. It is very important. Now, with respect to clinically meaningful for these patient populations, I think that we have so little that we offer them right now. They're largely treating wound areas. In the United States, we have 3 FDA-approved products for recessive dystrophic EB. We have none for EB simplex. And I would say that our products that are out there are helpful, but there's still room for improvement. And none of them target the inflammatory profile of these EB lesions, which is a really important niche here for treating with a new agent.
Thank you, Dr. Paller.
Yes.
Dr. Bauer, what are your thoughts on this question?
Yes, thank you very much. Dr. Paller has already pointed to the most important facts. Just want to add here that certainly for EBS, this is a big step forward for the patients with EBS. They are, as we have seen, 70% of all EB patients are EBS patients. They are completely underserved at the moment. They just have their bandages. They have some maybe some anti-pruritic agents in the future, but there's nothing on the market at the moment for wound healing, as opposed to recessive dystrophic EB, where there is 3 approved drugs in the US and 2 approved in Europe. So it's a big step for EBS patients, I guess, and it will be very good because at the moment we're talking about topical treatment for all forms of EB, and that's for recessive dystrophic EB. It's kind of an issue in the long run, I guess. I don't know what Amy has to say about this, because we're treating skin in recessive dystrophic EB, but we have several other complications in those patients. But certainly for EBS, the topical treatment will be very important in terms of meeting their needs. I think what is very important and interesting in the future that we would be able to combine treatments. We are thinking about already combining like genetic treatment with wound healing treatment for recessive dystrophic EB in different situations and different wounds, or combinatorial in terms of timing. And I think in the long run, if AC203 is approved for EBS and then probably for RDEB in the long run, we could think that there is another combination possible, because as Amy has said already, this is tackling the inflammation side of the whole pathogenic picture, and so that comes up with new features for treatments for clinicians. So and that's very good outlook for the EBS patients, but also for RDEB patients.
Can I add one other thing that I think is unique about the diasporan? We're not just dripping it into wounds where they're open. We're really treating the area, right? And I think for EB simplex, and hopefully also for RDEB, if there's anything we can do to increase the stability of the skin to prevent new blisters from coming up, we need to be doing that in both types, but particularly for EB simplex, given the major role of the fragility there, more than the healing itself. And that's what we are seeing here, is that we're not seeing just fast healing, we're seeing the prevention of new blisters coming up, which is unique. And I do want to emphasize that, in addition to totally agreeing with what Dr. Bauer just said.
Thank you. Michael, would you like to add any further perspectives from your end?
Thank you. Because first, just because by training, I'm not a really clinician here, but I definitely agree. In particular, the itching and the pain that actually are associated with those patients here, those are very important to treat, and to some extent, could be highly likely, linked to the inflammation, profile of the disease here. So I think Diasterin could provide a very unique angle, to treat, this type of disease here. Thank you.
Okay. Then let's go to the next question. Next slide, please. Building on that, we know that managing this disease remains incredibly difficult under current treatment protocols. What do you consider to be the key limitations and inadequacies of current standard of care treatments for EB simplex and RDEB? Dr. Peller, if we could start with you again.
Yeah, I think we covered a lot of these. There is very little out there, and we're largely just treating by using dressings and treating infections when they come up, especially with EB simplex, for which we have nothing. For the others, I think that one of the beauties of what we're starting to do more and more is not just use bland Vaseline, for example, on wounds. We're really starting to think about the pathogenesis of these diseases and how we can target elements of it. And we may not, as was mentioned, get everything with a single agent. We may have to be combining to be most effective. But a key limitation is the fact that when we're just using standard of care right now, especially for EB simplex, we are not really targeting what we need to target to either prevent the expansion or prevent the development, or to just help the field to stop having the inflammation and blisters that are happening right now.
Thank you. Dr. Bauer, your thoughts, your comments?
Yes, I totally agree with Amy. Let me say, we discussed EBS already. Let me say also for RDEB, the standard of care treatments that are approved these are most likely lifelong treatments. So that would be kind of the big inequity for those patients, because they would mainly looking for something, you know, applied once and healing forever. So we're talking about now transplantation, for example. If that, you know, work nicely, then this would be a very good feature for the patients. It does not work so 100%, so still have to work on that, even for our DB. So that's certainly something that has to be improved. Let me just quickly look into the chat, because there's already 4 questions. May I give the answers to that, or should I write it, or should we write it? I don't know.
Sure. I think we can. Well, we can jump to the Q&A.
Yeah, I'm not seeing anything there, so I'll leave that to you. Can I
Yeah.
make one other comment? I mean, we're talking here about a topical, and this is going to be really transformational for many of our patients. One thing to consider is that it's not just the skin. Many of our patients with EB simplex do have mucosal blisters. Certainly those with RDEB do as well. And I'd love to see, as we move forward, thinking about limitations that we'll still have, whether there might be some way to topically deliver anti-inflammatories, whether it's a nebulizer treatment, whether it's spraying something into the mouth in wound areas, just something to throw out there to consider.
Michael, any additional thoughts?
Yeah, I kind of would like to reiterate the point here, you know, because most of the current treatments here tend to be very supportive and in some ways a little bit more passive, you know, when the symptoms come, when the pain, itching, skin infection, we treat, you know, those symptoms here. So I think having a very active, you know, kind of the approach to the disease, you know, would really help. For example, you know, what Amy and Bauer, you know, mentioned, you know, if there's a way to stabilize the skin, to change the basic skin architecture or the microenvironment that actually makes the skin will react very different, you know, against mechanical trauma in the case of EBS, or for the RDEB, you know, wound healing things here, that would be actually really revolutionizing, you know, the current treatment.
Thank you. Then let's go to question number 3. Those limitations certainly highlight the urgent need for new therapeutic options. Fortunately, the landscape is evolving. With recent advances in EB treatment, how do you see these innovations being integrated into practice to improve real-world patient outcomes? Dr. Peller, could I turn it over to you first?
Sure. Well, we have several things out there now. They're all so different, right? I mean, we do have one topical that's currently applied. It's for junctional EB and dystrophic EB, not approved for EB simplex, although that's just because it really wasn't tested. I don't know if it would work or not. But the product we're talking about here, diacerin topically, is easy because as dressings are changed, or as is often the case with EB simplex, not everything is covered with a dressing because sometimes that exacerbates. It's easy to put something on in an area that might be affected once a day. So I think that will be easily integrated into real world practice. Certainly easier to integrate than major grafting procedures or even the time consuming getting a topical gene therapy dripped on. All of these are extremely important for our patients. We'll pick and choose a little bit, but we're also combining, as was already stated. So these innovations, I'll tell you, I try to, anything I possibly can for my patients, I will do. I see them easily being integrated into practice, very easy to put in a topical in day-to-day life.
Thank you. Dr. Bauer, how do you see this integration playing out?
Yes, I mean, there is, I guess, a very important feature also about diacerin. then that's not studied in the studies, in the clinical studies, but the preventative effect that Dr. Chan has already alluded to, that's something that would be really interesting, because if diasterin works as we think it works, it will stabilize the skin in the long run, you know. And so, and let me give you a small anecdote, because in the early days, we were giving diasterin to patients to try it out. And there was one guy who was always flying to South Africa to do golfing, and he has an 18-hole golf course. And he told us if he does the 18 hole without diasterin, he gets blisters. So he puts it on before he goes to the golf course. And if he puts it on before the golf course, he has no blisters. So that's kind of some real world stories we had already in the early days. That would be really an improvement for the patients when they foresee something, you know, severe happening to the skin, that they can start, you know, using diasterin and they don't get blistered. That would be a nice thing to see in the long run.
And that emphasizes, though, the fact that the diasterin is a topical that can get into the skin when it is intact, which is a major difference.
Right.
Others cannot get through the skin unless it is open and thus are applied to wounds. I think one of the important questions that's going to come up that you can hopefully address through the study already, in terms of at least some early evidence, is whether it will get through the bottom of the foot. Because this is an area that there are some people who really, this is the primary area of involvement with EB simplex in terms of affecting quality of life. And if the delivery through the bottom of the foot is effective, that is one other level of being huge. We didn't treat that here. We had to do we had to enroll patients who were more extensively involved, but hopefully we can get a clue and be able to think about that for the future as well. Another very practical, real world patient need.
Thank you. Michael, your perspective.
Oh, I'm certainly very excited about all this new advancement here. That means more advancement, more options, and more combination of options for the patients, you know. So that's definitely plus, you know. And when I remember, you know, back in the 1990s, when the genetics of DBS were, or dystrophic EB were discovered here, there was actually not too much talking about treatment, and we didn't even understand the disease at that time. The word inflammation was not even mentioned at all at that time here. So we came a very long way actually today to really make improvement in understanding the disease and all these treatments here. So I'm really look forward to the next, you know, few years, you know, all these options that can really available to the patients here, you know, maybe perhaps in a very customized, you know, kind of the, you know, combination of treatment, you know, for different types or different patients.
Next slide, please. That's a great transition into our specific efforts here at TWI Bio. We are optimistic about our clinical progress, but we also want to look at the broader clinical context. If AC203 is successfully approved, how do you foresee its impact on shifting the current standard of care for EB simplex? Again, Dr. Pala, could I have your thoughts first?
Well, I think it could be a game changer for our patients with EB simplex. It's certainly looks great in the trials that we've seen so far, and it's going to be no extra work for them to be putting this on their wounds or under dressings, because they're already putting on a moisturizer. They're already applying to wounds. They can just do this instead. So I really see it as being something that not only can they apply to wounds, but as we were just talking about, if it prevents wounds, it's going to totally change what they're able to do in life, which helps their quality. Hopefully will also make them feel better, less pain, less itch, and certainly maybe fewer blisters from what we can see. So I can't imagine why we wouldn't be using this as our go-to agent hopefully as the first approved therapy for EB simplex.
Thank you, Dr. Paller. Dr. Bauer, what impact do you foresee?
Yes, so there's nothing to add to this comment. It will change the standard of care completely. It can be integrated in the daily practice or in the every other day practice, depending on what the patients do with their wounds. It's easy. It's basically without side effects, and it can be distributed all over the world. Basically, it's nothing very complex as we see it for dystrophic EB, for example. So it will be very interesting to see how this will fare then in the long run in those patients.
Thank you. Michael, please feel free to add your perspectives.
I definitely look forward, to the outcomes here, and I think it could potentially. It is that I agree with Amy. Definitely, it's a game changer. For EBS, it's not just the first, to be approved here, but the underlying mechanism, how it works, and potentially how could it prevent, blister formation here. Those are very important, aspect for the patients here, and it can also potentially open the door for other indications as well. So I think this is. Yeah, very remain optimistic. Thank you.
Mm-hmm. Let's go to the last question. It's certainly our hope that AC203 becomes a cornerstone of care, but we are also thinking about where else this mechanism of action might be beneficial. Looking beyond generalized EB simplex, what potential do you see for AC203 in other EB subtypes or related inflammatory skin disorders? Dr. Paller, if you could start us off.
Sure. Well, we have several questions here. I see that Dr. Bauer was referring to earlier about dominant dystrophic EB, junctional EB, and as was discussed here, recessive dystrophic EB. And I think we're seeing more and more transcriptomic, proteomic analyses of a wider variety of the different types of EB. And certainly if there's evidence of increased in IL-1 beta in these other forms, it would make a lot of sense to move forward, or even just some small pilot studies to take a look at that as well. I'm excited about it for other inflammatory skin diseases. So we've got a keratinopathy, keratin 5, keratin 14, but there are other keratin gene disorders that we as dermatologists see. I see a lot of patients with what's been called epidermolytic ichthyosis that are from a variety of keratin gene abnormalities, and we think that the mechanism is very similar. We already know there's an increase in interleukin-1 beta in these disorders, and this is a topical that could be applied widely, and not only would reduce the skin fragility in that disorder, but maybe over time could reduce the development, if we could treat early on in life, of this thickening of skin, or even reverse the thickening that characterizes this disorder in older individuals. And similarly, there are others, like another disorder with thickened soles of the feet and blistering that is tremendously incapacitating in terms of quality of life, again, particularly in summer months, which are often a time that are harder for our patients with EB simplex. So those are two examples of non-EB disorders that from a pathogenesis-based situation might lead us to think about using this agent.
Thank you, Dr. Paller. Dr. Bauer, do you have any comments?
Yes, so that's also kind of an answer already to the comments in the chat. Certainly, we will have a look into junctional EB, but first we have to finish the studies with simplex and have to move forward with recessive dystrophic EB. And then we will have a look into junctional EB because there is inflammation, as correctly pointed out. And there is inflammation, there is IL-1 beta, I guess. And looking into the wounds of the patients, they are very similar, actually, of junctional and dystrophic EB wounds. So it might be working, but we have no data at the moment. This dominant dystrophic EB is another issue. There's less inflammation, I guess. This is just a missense mutation with the structural deficiencies, but there might be also inflammation involved, less than in recessive dystrophic EB. So we have again not studied this so far, and in the long run we have a look into it. So that's basically indications that we can look into in the EB area. Amy has pointed out all the keratinopathies. It's a wide area. Would be great to have some data on efficacy for diasterin there. And we have actually studied psoriasis, a mouse model of psoriasis, and we have seen some moderate effects there. And that's something we have to discuss also. That's an issue. But although certainly it's known that IL-1 beta does not play a major role in psoriasis.
Thank you, Dr. Paller and Dr. Bauer. Michael, any further thoughts?
I think I can just a quick comment, from the preclinical perspective here, because diasterin has been known to have anti-inflammatory properties, and IL-1 beta is one of the major pro-inflammatory cytokine that actually diasterin target here. But it definitely has a very unique, inflammatory signature here. So I think in the future, if we can better understand, the anti-inflammatory signature of the diasterin here. For example, we have published that actually diasterin can also suppress, TNF alpha, in macrophage here. So I think that would actually give us a much better idea in the future, what type of, skin or relevant, related inflammatory skin disease could be, applicable, for diasterin here. So thank you.
Thank you all for this incredibly insightful discussion. This concludes the formal panel portion of our webinar. Next slide, please. As we bring this webinar to a close, I want to leave you with a few critical take-home messages. First, our target for the AC203 readout unblinding is Q4 this year. Following this unblinding, we will finalize and announce the detailed timelines for our regulatory consultation with both the US FDA and EMA. Furthermore, we are very encouraged that diasterin improved wound healing in all patients treated under the named patient program. Moving forward, we will be collecting further data to evaluate the potential therapeutic benefits of AC203 in RDEB. Next slide, please. This work requires immense collaboration, and I want to express our deepest acknowledgments to everyone involved. Thank you again to Dr. Johann Bauer, Dr. Amy Paller, and Dr. Michael Chen for participating in today's discussion. Most importantly, we want to thank the EBS patients, their families, and the principal investigators for their vital participation and support in our EBS studies. We also extend our sincere gratitude to Dr. Verena Valley at the University Hospital Salzburg, Dr. Mary Spellman at Castle Creek Biosciences, Deborah of America, and Dr. Tomoko Chubachi for their invaluable contributions to this program. Next slide, please. Advancing this research is truly a worldwide effort. As you can see from the faces and locations on this slide, our global clinical sites span across the United States, Australia, Europe, and Asia. To all the dedicated teams working tirelessly at these sites, thank you again for your unwavering commitment to our clinical trials. Next slide, please. Finally, our mission at TWIB is anchored by our continued partnerships with DEBRA organizations around the world. Whether it is participating in the DEBRA Care Conference, supporting DEBRA UK's focus on the future of EB research, or engaging at the annual ESDR meeting and Deborah Italy's EB Day, we are dedicated to standing alongside the global EB community. Thank you all for your time, attention, and shared dedication to improving patients' lives today.
Should we go through the chat questions? There is one from Sophie Kitzmüller from the EB House, actually, that goes to the company. Will NDA submissions be done as soon as data from the EBS trial are available, or will you wait for data for RDP as well to have a broader indication? I guess that's for TWRB.
Yeah. Now let's move on to the final segment of our agenda today, the Q&A session. We want to hear from you to address. I've got several questions on my screen. The first question is about when and where will the topical treatment for EB simplex be launched? AC203 is the fastest drug under development for EB simplex, and TWIB will have a much clearer timeline for when the treatment will be available after we analyze the results from our ongoing EB Shield study in Q4 this year, and then consult with regulatory agencies, including the US FDA and the EMA. And I got a question about daily care. What are the best ways to control itching and manage wounds in EB, especially in young children? Could I refer this question to both Dr. Palo and Dr. Bauer?
Well, I guess I can say a little bit. Hopefully, as we find agents like this, and maybe Johan can address a little bit more the impact on itch in his experience here. But as we find products like this that are anti-inflammatory, I think that they will also reduce, and that also overall reduce the blister burden and the inflammatory burden. I think that we'll see that helping very much. I would say that this is a problem because the itch in EB is not related to histamine. So antihistamines that we've used for a long time have not been terribly helpful, other than sedating and helping people sleep at night, that sort of problem. We've not seen in general with itch that agents like gabapentin do a whole lot. They help much more with pain. There's been some early work with some of the inhibitors like serlopitant that suggest that they may be helpful, not really used for EB simplex. In our trials of a commercially available injectable biologic drug that's available down to 6 months of age, called dupilumab, which targets a different inflammatory molecule in a particular arm of the immune system. We've had patients with EB simplex whose itch has been dramatically reduced, and a fewer numbers that it hasn't been helped. So that may be something to consider in the future. But again, if a topical agent can be helpful, especially with EB simplex, I think that would be highly valuable. But I'd love to hear from Johan a little bit about his experience with itch with this product.
Yeah, so we have studied itch and pain alongside always with the blister counting or the other features of the wound. The effect in our hands on itch was less pronounced, I have to say, but this was just in those 2 small studies. We will looking forward to see what the effect will be in the bigger study coming out in October. So probably again we can have a combination, you know. So if the reduction of pruritus of itching is not enough for the patient under diasterin, we might have to add something like dupilumab in addition, and so that should then suffice to have a good effect on the quality of life of the patient.
Yeah, and there is a study ongoing that is going to move forward towards a label indication for EB, for itch specifically, of dupilumab. That's moving forward. And there are other anti-itch agents that as the age comes down to that very important childhood period, we'll see if those could be helpful.
Thank you. The second question is also clinical. How close are we to a cure for EB, and what long-term complications, especially kidney disease, should patients be aware of?
Are we talking about EB as a whole, or are we talking about EB simplex? Yes.
The audience member didn't specify.
All right. I'll talk in some generalities. So to cure AB, we would probably, we'd have to have some kind of a genetic approach that permanently alters the cells and their expression of normal versus abnormal gene products. That's a very high bar. The farthest along with that is gene-corrected keratinocyte or outer skin cell grafts that have become available for recessive dystrophic EB. And it's complicated because you wouldn't just put anybody in that. Of course, everybody would love to have a cure, to have their skin totally altered, but it's a difficult prospect. You do have to take off large areas of skin to put on these rectangular grafts. The amount that one can use at any one time is limited, and it's by no means 100%. Looking for looking for improved quality of life, reduced pain, reduced itch, and areas that don't blister as much. There is absolutely nothing right now for any other type of EB that comes close to cure. But there's a lot that we can do that's not cure. If you look at common inflammatory problems like eczema, psoriasis, we're not curing them, but we do have highly effective agents that would target the itch, would target the inflammation. And so for the next several years, that's really where treatment's going to be. And while we would all like cures, I would love to see highly effective topical or systemic treatments that just reduce the burden for patients and families.
Dr. Bauer, any comments?
Yes, so I just want to add that it's feasible. The cure is feasible. Depends on the form of EB at the moment. We have been treating one patient with junctional EB with not severe form, intermediate form, with 80% transplantations basically all over his body, and he's leading a normal life. That comes close to cure for me. For several reasons, we could not proceed with more treatments like this, but it gives us the idea it's possible for specific forms of EB. It will be tough for recessive dystrophic because transplantations are more complicated there, and you have the internal complications, which then it's not a cure if you transplant the skin. For EBS, we don't think we will have transplantations, and so we will be stuck with local therapies that are hopefully good enough in the long run. So it depends on the form. As I said, it's possible in some forms, but we have to work in others.
Thank you. So it looks like we are approaching the end of our scheduled time. If we did not get to your question live today, please rest assured that our team will review the remaining submissions and follow up with you directly. On behalf of everyone at TWR Bio, I want to extend my deepest gratitude to our incredible speakers, Dr. Chen, Dr. Bauer, and Dr. Paller, for sharing their invaluable time and expertise. Most importantly, thank you to all of you in the audience for joining us across different time zones. As I mentioned at the beginning of our session, our mission is driven by the core belief that every patient and their family deserve a better life, and your participation today brings us one step closer to realizing that goal for the EB community. A recording of today's webinar will be made available shortly. Thank you again, and please have a wonderful rest of your day. Thank you very much.
Thank you very much for
Thank
your
you.
time.
Thank you.
Thank
Bye-bye.
you. Bye-bye.
Bye.